Welcome to Tech Tuesday, presented today by the Health Technology Forum. I'm Pete Poorman, Director of Corporate Relations at UT Dallas and a Tri-Chair of the Health Tech Forum Steering Committee. I'm pleased to open today's presentation. On behalf of all of Tech Titan's forums, I hope you are enjoying this Tech Tuesday programming, which showcases our 8 technology forums through cutting-edge presentations, like the one being brought to you today. Tech Tuesdays, which occur on the second and fourth Tuesdays of every month, both educate and generate discussion around those topics among the tech community. So mark your calendars now for the second and fourth Tuesday of every month during the noon hour for Tech Tuesday and be part of this new and growing program series. And a quick mention to announce the next Tech Tuesday program on May 11 will be presented by the Blockchain Forum. The program will feature a panel discussion on the realities of digital currency by representatives of Deloitte, Silicon Valley Bank, and the Dallas Federal Reserve Bank. You may find additional information and register for this program and all Tech Titans events on the website at techtitans.org. Today we're using the Remo event platform. You may ask questions during the presentation using the Q&A feature located to the right of the main stage. Please put your questions in the Q&A and not in the general chat. That way, our program moderator, can monitor those and provide them to the speakers. Following the program, you're invited to remain on the call for an optional networking session. This gives us time to meet each other, and we'll run from 1 till about 1:30. So on to our program today. Our program today is about the Defense Advanced Research Project Agency, or DARPA. The agency has developed a pandemic prevention platform to support military readiness and global stability as part of the COVID-19 pandemic response. To introduce our keynote speaker and moderator discussion today is Kim Mayer, the Associate Vice President for Research and Interim Executive Director of the Center for Entrepreneurship and Economic Innovation at UT Arlington, and a member of the Health Tech Forum Committee. Welcome, Kim.
Thank you, Pete. I'm really excited to be able to introduce Dr. Amy Jenkins to our group today. Amy has been a program manager at DARPA since June of 2019, so she's coming up on her 2-year anniversary as a program manager. At DARPA, she focuses on the development of platform technologies to combat infectious diseases and on novel manufacturing methods that will allow rapid response if and when those diseases threaten our population. Before joining DARPA, Amy was a senior scientist at Gryphon Schafer, where she worked on the development of programs that target infectious disease threats, including those of interest to DARPA's Biological Technologies Office. So she has a longer history with the BTO and some more awareness that she'll be able to share with us today. Amy's also worked at MedImmune, and she served as a National Research Council postdoctoral fellow at, get this, the U.S. Army Medical Research Institute of Infectious Disease, USAMRIID. So for any of you hot zone enthusiasts out there like myself, she actually worked there and gained expertise working on virulence and multi-drug resistance in bacterial pathogens while she was there. She received her PhD in chemistry and chemical biology from Cornell and her bachelor's in chemistry and biomolecular science from Clarkson University in New York. So I'm really looking forward to hearing your talk today, Amy. Please tell us all about DARPA's role in developing these novel technologies to prevent and prepare for pandemics.
Thank you, Kim, for the great introduction. I'm very excited to be here today and to be able to tell you about the types of technologies that we've been investing in at DARPA and why we're even investing in these technologies in the first place. So always very excited to share the types of things that we're doing. And we're excited that we've been able to make an impact on this pandemic and hopeful that we'll be able to do even more in the future. So as Kim mentioned, today I'm going to talk about the pandemic prevention platform and the role of technology innovation in preparedness. So on the next slide, what I'd like to do is just give you a brief overview of DARPA, just in case you're not familiar with DARPA. So some of you may be quite familiar and some of you may not, but DARPA was funded over 60 years ago in response to the Sputnik satellite. So as many people know, Russia launched the Sputnik satellite, and it was a very trying time here in the United States because we felt that, another foreign adversary had beat us to the punch on something. And it was scary, right? The thought of something that was in the sky that could monitor us and potentially launch missiles at us was very frightening. And so at that time, President Eisenhower launched the Defense Advanced Research Projects Agency, at that time just known as DARPA, or the Advanced Research Projects Agency, to prevent surprise. He said, we'll never be surprised again. And the kind of colloquial terms we use at DARPA are we say that we're here to prevent and create strategic surprise. And so DARPA really invests in technology that is meant to be high risk, high reward. So we're kind of out there on the leading edge of the R&D ecosystem. We invest in things that a lot of people laugh at us for and say that's not possible. You're breaking scientific rules. There are reasons that this won't succeed. And we typically say, okay, you're right. There probably are reasons this won't succeed, but there are reasons that it may succeed, and we won't know unless we try. And so we're very lucky as an agency to be able to try to push those boundaries. So on the next slide are some examples that you all may be quite familiar with. Sorry, there's an animation here. So on the next slide, if you're familiar with GPS technology, of course, this is kind of something that has been built out of some DARPA programs, and the ability to miniaturize this and put it in something like handheld, like your iPhone or something that you can have in your Garmin in your car. Just one of the examples of some of the types of technologies that have come out of DARPA. And I think, a lot of times what the standard is that DARPA has created the internet and certainly laid the seeds for the modern internet as we know it. But as you'll see on the next slide, we are a defense agency. So there's the D in front of DARPA, stands for defense, of course. And so our main goal is to develop technologies that provide readiness and make our military members more mission ready, mission capable, and ensure their safety in vulnerable situations. And so you may ask, why does DARPA and why would a defense agency like DARPA be so interested in funding things that are involved in pandemics? Isn't this a homeland security initiative? And of course, we do work across our interagency and with other organizations as well. But three things that I want to point out as to the role that DARPA plays in infectious disease and pandemic response, and the reason that we're involved in this in the first place. The first is that, of course, as you know, the DoD deploys military personnel around the globe. So we have a worldwide deployed military force where these military personnel may encounter infectious disease threats that we wouldn't encounter here in our homeland. So a lot of different infectious disease threats, think of things like dengue and malaria. So we have a great interest in keeping those military personnel safe from infectious diseases and from the diseases that these pathogens can cause. Second is that while we are a worldwide deployed military force, we also are very much play a role in stabilizing communities. And part of that is humanitarian missions. And so this may not be something that is quite as obvious, but the DoD does respond oftentimes to humanitarian missions. And so one that I really like to point out is that the Department of Defense deployed almost 3,000 military personnel to the West Africa Ebola outbreak in 2014. They were deployed largely for operations and logistics, but clearly we put military personnel in harm's way in an Ebola outbreak zone and in the middle of a pretty severe endemic situation. So we really want to enable technologies that allow the DoD population that we're going to put in harm's way to be safe. But additionally, we want to be able to bring with us those types of technologies that may help stabilize a particular region. And so while we want to keep the military safe, we also want to keep civilian population safe, because that helps stabilize and can play a very important role in humanitarian missions. So that's kind of the why as to why DARPA is involved in infectious diseases and pandemic response. And I think we all know now that the Department of Defense has played a role in this current pandemic and has been impacted by it. And there's been many known examples within the media, including the USS Theodore Roosevelt, and how the pandemic can actually impact a warfighter readiness and have an impact on the ability for us to immediately deploy. And so on the next slide, I'd like to just talk you through some of the early investments that DARPA has made that are being deployed now. So approximately a decade ago now, DARPA began investing in nucleic acid technology. And I will note that DARPA personnel do not stay at DARPA forever. So we are on term appointments. That's on purpose. It's meant to keep us on our toes and keep us focused on the prize and the thing that we want to do at hand. And so I did not start these investments, and worked with some really great previous program managers at DARPA that started many of these current investments. But there was very much an interest about a decade ago to say, what could we do in the infectious disease realm? I mean, there are emerging technologies now, but really the problem with the Department of Defense, and the problem that we saw at that time, was that we didn't have months or years to respond to an infectious disease. I think, in 2011, around the time 2010, 2011, when we started investing in this technology, I think the fastest that we had ever developed a vaccine was approximately a 4-year timeline. And that timeline is much too slow if we have military personnel that we want to deploy in a, basically moment's notice. It would really be great to have technology that would allow us to make vaccines or therapeutics much faster. And so one of the program managers at the time actually said, well, one thing that could be faster is if we made these modalities as nucleic acids. Nucleic acids, at that time, it was believed, and I think it's been proven, that they have a significant manufacturing benefit. We can just make them a lot faster than we can make our more traditional vaccines and therapeutics. So we began investing in that technology, and what we called it is using the body as the bioreactor. So rather than growing up big vats of vaccines in large bioreactors, or rather than growing up big vats of antibodies that are used to treat diseases in large bioreactors, we actually thought, well, could you give the code for those proteins, the DNA or the RNA code? Could you inject that into your cells and let your own cells make that for you? Turn your body into the bioreactor. Now, a decade ago, this was quite a novel concept. And I think a lot of people, particularly at that time, we would spend a lot of time on this slide because people just fundamentally were like, wait, I don't understand what you're doing here. Fortunately or not, I think people are much more familiar with the use of mRNA technology now, because it is currently, of course, being deployed in this pandemic and is part of the Moderna and Pfizer vaccines. And as you can see here, I have a little call out for Moderna, because DARPA was one of the early investors in Moderna technology and began funding Moderna in 2013 to evaluate the potential for using nucleic acid-based technology for both vaccines and antibodies. And I'm proud to say that I was able to get my Moderna vaccine just Friday. So it's pretty exciting to see the use of this technology come full circle. And I think it has been proven out to some degree that, of course, nucleic acids can be manufactured faster. And we're the first, of course, to be deployed in this pandemic response in the United States. So we were excited to be involved in that. But we wanted to take it one step further, and not only just focusing on vaccines, because vaccines, as we know, provide us long-term protection. But as you all are very well aware now, when you get your shot of vaccine, you have a couple weeks and you got to go back 4 weeks later and get your second shot. And then you've got about 2 weeks after that before you're fully protected. So in most situations, we're looking at anywhere from 2 to 6 or more weeks before you're fully protected from a vaccine. And remember what I said just a few minutes ago. Sometimes we deploy military personnel with very little notice. We don't have 2 to 6+ weeks of lead time. So while it's great to be able to give that vaccine at the time, and it will eventually kick in, if you will, we wanted to also invest in some technology that would allow us to provide immediate, maybe temporary, but immediate protection to cover that intervening time. Is there a shot we could give today, and by tonight, I'd be protected? And so that's where we began developing the pandemic prevention platform. And so I'll give an overview of this on the next slide. And this is just kind of a schematic overview of this program. And what we were aiming to do with this program was to develop a platform that would allow us to take any pandemic virus, any virus of pandemic potential, or once we knew there was a virus that was causing of concern or potential for causing a pandemic, could we initiate this platform and in under 60 days develop approximately 20,000 doses of this treatment to prevent the pandemic. So I use the word treatment to prevent. That is a little bit misleading. What we really are looking for is a drug that prevents. So very similar to a vaccine, but not quite a vaccine. And then it doesn't last a long time, but it does prevent disease. And the technology that we felt was the most likely to be able to meet these metrics was a combination of two technologies. The first was the use of monoclonal antibodies. And you'll see that in these kind of middle pictures here, finding antibodies, evolving antibodies, essentially making sure you have great antibodies. We liked monoclonal antibody technology. The reason that we were focused on monoclonal antibodies is because they are kind of very cool molecules. They are molecules that can both prevent a disease and treat a disease. So you have that benefit that they could be used for both. They are proteins that are natural in your body. Your body, if you got your vaccine, your body is making them right now against SARS-CoV-2. So they exist naturally in people, and they last for a fairly long time as opposed to a small molecule like an antibiotic you may take. You know, when you think about it, you take an antibiotic and you probably need to take it, 3 times a day for 10 days, something like that. An antibody, sometimes you can engineer them and they can last for anywhere from 3 to 6 months with one injection. So that was very attractive from DoD personnel standpoint, because not only could it be immediately protective, but it could potentially last for quite some time to either cover an entire deployment or to cover the time before a vaccine kicks in. So that was the first half of the technology we were interested in developing. The second half of the technology was, well, what can we do to deliver it very quickly? And this is where we said, well, we think one of the ways to do this very quickly would potentially be to use that RNA or that DNA technology that we were so interested in the vaccine space. Because again, there's a manufacturing benefit there. And so we started this program in 2017. And then in 2019, as the manager of a program called the Pandemic Prevention Platform, you can imagine things got quite interesting. And we did utilize this platform to respond to the COVID-19 pandemic. However, I will say that at that time, the program was only halfway done, about 2 years into a 4-year program. So this platform, as you see it here, wasn't quite fully ready to deploy against the COVID-19 pandemic. But that middle piece of finding and making the antibody as robust as possible, it was ready. And so we were able to initiate this platform for the COVID-19 pandemic. And so on the next 2 slides, I'll give you some examples of what we were able to achieve with this program during the pandemic. So the first one I want to talk about is a small company that we funded under this program called AbCellera Biologics. And some of you may be familiar with them even now. I think very few people would have been familiar with them just over a year ago, but some people may have heard this name now, and they are partnered with Eli Lilly. But this group actually began screening some of the blood samples from patients that had recovered from SARS-CoV-2 very early on in the pandemic, some of the first patients. And those patients that recovered, of course, in their blood, they had antibodies that were protecting them or that helped them recover. And so, of course, under fully informed consent and all of the things that you do under clinical research, those patients donated their blood and allowed companies like AbCellera Biologics to start looking in their blood to find some of those antibodies that were the best ones. And so this is an example of the timeline that it took AbCellera to identify those antibodies. And as you can see here, they started the screening process on day 4. And by day 23, they had chosen an antibody to move into advanced development along with Eli Lilly and partnering with Eli Lilly. This is, for those of you that may be very familiar with the antibody discovery space, is just lightning speed. As Kim mentioned, I came from MedImmune. I was working in a company that worked on antibody technology. And prior to this type of investment, it would have taken anywhere from 6 months to a year to find antibodies traditionally. And we were able to do it in 23 days. And of course, AbCellera partnered with Eli Lilly. Eli Lilly was not funded under this program, so I should note that they were not government funded at this point. But they were able to move into the advanced development. And on 90 days, on day 90, they began testing these in COVID-19 patients. So they entered phase 1 clinical trials. So not quite 60 days, but 90 days. And we were very, very proud of the ability to start making a difference in patients' lives within about 90 days. And then on November 9th of 2020 was a big day. And that was the day that this antibody that many of you may know as BAM now was issued an emergency use authorization and was used in to treat COVID-19 disease in mild to moderate patients. And so we're very excited that this platform was able to be utilized in this pandemic. So on the next slide, there are some results for a similar approach by our researchers under the same program at Vanderbilt University and at AstraZeneca. And so this is a very similar story in that Vanderbilt University also began doing this process. So all of the teams in this program essentially began this process when we realized that there was a pandemic. Vanderbilt began a very similar process of identifying antibodies. And they actually entered into a licensing agreement with AstraZeneca, who was a fellow P3 performer, as we call them, but a P3 grantee, if you will. They entered into a licensing agreement together for their antibodies that can be used to both prevent and treat COVID disease. And these antibodies are currently in advanced late-stage clinical testing for both the prevention and the treatment of COVID-19 disease. The particular benefit here is that this product is a simple intramuscular injection. So you may have heard about some of the problems with antibodies, and just IV infusions can be difficult in a pandemic setting. And we're very excited that this product would be an intramuscular injection, somewhat similar to a vaccine. So very excited about the potential for this product as well. So that's kind of what DARPA has done in the pandemic prevention realm to date under this program, and what we did in the COVID-19 pandemic. I certainly am summing it up here. There are a lot of other efforts, and I'd be happy to elaborate on those at some other time. But I do want to touch on another program that we have, which is maybe a bit more out there in terms of DARPA technology. So this program is a slightly more advanced program, of course, because it's moved into clinical testing and is being used in patients now, would be considered quite an advanced program for a DARPA program. But we do have other efforts that are very early stage, early in their R&D landscape. And as I mentioned at the beginning, DARPA does typically function very far out in that R&D, at the edge of that R&D landscape and the technologies that are very new. And while the technologies that I've discussed here for the pandemic prevention platform are still quite on the edge, there are technologies that are even newer than this and very exciting additionally. So on the next slide, I'll talk about this new approach. So this is the up and coming approach. And so in this approach, we are actually aiming to use the code that's written in your DNA to protect you against threats, to protect against chemical or biological, or radiological or nuclear threats that DoD personnel may encounter. And the way we're doing this is by delivering a certain type of medical countermeasure, which is what we generally call our vaccines or therapeutics, delivering a certain type of medical countermeasure that may allow us to turn on the protection that is already within your body. So as we already know, many people are infected with SARS-CoV-2 and they get sick, or some of them don't even get sick, and of course they recover. And there are mechanisms within our bodies that allow us to recover from that. Of course, unfortunately, sometimes people are not able to recover. So those mechanisms aren't tried and true. They don't always work. But the thought with this program was, well, if those are in your body and you just have to get them to work just right, maybe we can develop technologies that allow your body to ensure that they're going to work just right in the situation that we may need. And that's what this program, called the Prepare program, aims to do. And so I'll walk you through a little bit of what this technology actually is on the next slide. And so here, you may have heard about CRISPR-Cas9 technology, and this program does rely on CRISPR-Cas9 type technology. You'll see over here on the left-hand side of this slide, and I want to be very clear that what this program is not doing, it is not editing human DNA in any way. So cutting of human DNA is out of scope of this program. This is not what we're using. But what we are using, so this CRISPR-Cas9 technology that you may have heard about is a very, very somewhat new emerging technology that we've just identified, I should say, as new, but an exciting technology that has two components of it. And I many people likely know this, but these components are both the Cas9 enzyme and the guide RNA or gRNA. And so the guide RNA actually works as what we like to call it. It's essentially a GPS for your genome. It says, or for a DNA, it says this is where you need to go. And then this Cas9 is an enzyme that actually cuts DNA. And so these taken together can be a very powerful tool. But what we said in this program was there are emerging analogs of this Cas9 called dead Cas9 or Cas13. They don't cut DNA, they're dead. That's why they're called dead Cas9, very creative labeling there. But what they still do have is that exquisite ability to hone to a very particular part of the DNA. They still have that GPS capability. And so we said, well, maybe we can use this dead Cas9. It no longer cuts, but it can take us right where we want to go in the genome. And so perhaps if we knew a certain area of the genome was where you had the potential for protection against a particular threat, so maybe we know that this gene, if it's turned on or turned off, can prevent you from having some of the deleterious effects of radiation sickness. Could we go in and very specifically target that gene and turn it on, for example, and then maybe not have those deleterious effects of exposure to radiation? Or similarly, could we, if we knew that a gene was involved in preventing the effects of a chemical threat, could we go in and regulate that gene and turn it off or turn it on so that we don't have some of those effects? And so that's what we're doing in this program. We have two approaches. The first is the host focused, and that's what I've been describing largely here, where there are genes in your body that can provide protection. They just may not provide enough protection. So we can give them a little bump by helping them out with this technology. Or we have a second part of the program called the virus focused part of the program. And so you may have remembered I mentioned Cas13A. So this is a different type of these enzymes. It's in the same general family, but rather than targeting DNA, it actually targets RNA. It actually cuts RNA. And as you all are maybe familiar with now, viruses like SARS-CoV-2 and influenza, they're RNA viruses. They contain RNA. And so the thought here was, well, if Cas13A cuts RNA, can we make it cut viral RNA? Can we use it so that it doesn't target the host, because it can be very, very specific, and we make it specifically target SARS-CoV-2, for example, and essentially shred up the SARS-CoV-2 before it can make us sick. And so those are the two focused approaches that we have in this program. And I can't reiterate enough that we are not editing humans and we're not cutting human DNA. But what we are doing is developing a technology that would be non-permanent. So you could use it and it will last for as long as we want it to last. So we want it to be very tunable. We want it to be reversible and transient. So we don't want this to be something that's consistently upregulating those genes or consistently downregulating them when we have no way to turn them off. It's very important that it be reversible and it only lasts for as long as we want it to last. We're leaving the genomic DNA intact. And we want to be able to make this very modular. So you could think of a situation where you could have these dead Cas9 enzymes. You could have a lot of them made. And all you have to do is go make these guide RNAs, essentially your little GPSs. And these guide RNAs are very small. They're fairly easy to make. And so if we had all these dead Cas9 kind of stocked, then all we may need to do is just make that little bit of guide RNA when we know what our new threat is, and we could very quickly respond to new threats, whether they be chemical, biological, or radiological, nuclear in nature. And so we think that that may significantly decrease the timeline, even based off the 60 to 90 days that we're currently targeting. We may be able to go even faster than that with this technology. So I know that this may sound quite science fiction, and you say, okay, you're going to put this crazy enzyme in that's going to upregulate or change the expression of a gene that then somehow has a downstream effect that prevents you from getting infected. And this sounds like some giant robo rally like thing that you have to put together and try to get this all to work at once. So is this going to actually work? And I think that was the question at the beginning of DARPA programs, and a very important question that is usually posed at the beginning of DARPA programs. But the good news is that we are about 2 years into this program now, and we do have very promising data. So on the next slide, I'll show an example of some of that data. And this is from a paper that was just recently published in Nature Biotech. And this is from our performer at Georgia Tech University. Phil Santangelo is the PI here. And what Phil was actually able to show was that in hamsters that were infected with SARS-CoV-2, hamsters that were uninfected. So hamsters, the way we tell if they've got SARS-CoV-2 is they don't eat and they don't gain weight. They kind of stay at their standard weight. Hamsters that are healthy and happy, they gain weight. So as you can see here in black on the uninfected line, hamsters that were not infected with SARS-CoV-2, they gained all their normal amount of weight. Hamsters that were infected with SARS-CoV-2 but were not treated at all in the red, they basically just gained a tiny bit of weight, and they actually initially lost weight, which is kind of the standard for hamsters in this model. Whereas you can see in the green line, hamsters that were infected but given this CRISPR-based antiviral essentially gained as much weight as the uninfected hamsters, showing that these hamsters were protected from this infection. And then we had the appropriate controls where we said, well, we'll just give them CRISPR, but we won't target it toward any point of the genome, or we won't target it toward the virus. We'll just give it to them and just make sure there's not a placebo effect, if you will. And there wasn't. Those hamsters did not gain any weight or gain very little weight. So we were very excited to show in a fairly large animal model, that's a well-established animal model for SARS-CoV-2 infection, that these types of antivirals can actually be used to treat animals and to prevent them from getting SARS-CoV-2 disease. So very exciting early data. Of course, we're only currently doing this in mice and hamsters, small animals. We're looking to move to larger animals shortly. And what this program won't do is move into clinical studies. So right now, DARPA is just investing in establishing the technology, making sure it's safe, understanding everything we can about its mechanism of action and how it works. But we're hopeful that this may be a modality that would be deployed in the future, and maybe even against the next pandemic, dare I say it. So that is kind of where we stand with these novel technologies to combat the next pandemic. But then I do want to touch on one last point, and this relates to the ability to actually make all this stuff. As you all have heard, there's a lot of issues around just making this and manufacturing it. There's issues with these large facilities having the capacity. Do we have enough vaccines? Are we able to get them where they need to be? This is something that I think is a forefront of people's minds now, and was certainly a concern of mine even before the pandemic. And so on the next couple slides, I just want to talk about what we call distributed manufacturing. And this really, the goal here was to enable a different type of manufacturing modality. As you know, we're manufacturing in very large centralized facilities now, and we ship our vaccines and all of our therapeutics around the country and around the globe for then ultimate distribution. And DARPA has said, wouldn't it be interesting if we didn't do that? I mean, we're not looking to replace that entirely, because you cannot manufacture enough for an entire globe in a distributed setting. So we're not looking to replace the investment that we've made in large scale manufacturing. That still absolutely has a place. But in particular, for an organization like the Department of Defense that often responds to humanitarian missions that are somewhat smaller in scale than a global pandemic, wouldn't it be great if we could take the ability to manufacture whatever we may need there on demand? The example I always used prior to the pandemic was the 2018 Ebola outbreak. So you may not know that there was a 2014 Ebola outbreak that we were all very familiar with, but there was another one in 2018 in the Democratic Republic of the Congo. And similar to West Africa, a lot of villages that were very hard to access. And oftentimes, although the Department of Defense did not respond to the one in the Congo, and they did respond during the West Africa outbreak, the Department of Defense may be called upon to help. Another example is the Department of Defense was called upon to help after the Haiti earthquakes, in which there was a cholera outbreak. And so wouldn't it be great if we could make what we need on demand right there in a small footprint that we can take with us, make the 10,000 doses that we may need, not the 7 billion doses that we may need for the entire globe. So on the next slide, I'll just give you a brief overview of this new program that I started fairly recently. And this program has just kicked off, so we don't have a lot of results, but we're very excited about it. And the program actually was thought of. The announcement for the program went out, and all of the kind of discussions of the program occurred before the pandemic, and then the pandemic hit. And I think it's been eye-opening how important this will be. But the goal of this is to leverage some of our medical countermeasures pipelines that we currently have. We have these investments in all the things I told you about before, in antibodies and in nucleic acid-based vaccines and things like CRISPR-Cas9 that just use these little tiny guide RNAs. Can we utilize that, and can we now develop a technology that will allow us to manufacture these from basic raw materials, essentially code, so the A, T, C's, or G's that DNA and RNA are made up of, basic raw materials, and can you synthesize these on demand, actually fill them vialed and all, and can you do this in a setting that is austere, and can you do this wherever you need to do this, and can you use that then to protect warfighters and civilian populations in these regions? And so on the next slide, I'll talk a little bit about what we're thinking with this technology. And so the goal was to actually develop essentially a system. I don't like to call it a device because that would imply that it's like maybe the size of a little PCR machine or something, but a bit of a system. The metrics we set out in our broad agency announcement were 6 feet by 6 feet by 6 feet. So could easily fit in the back of a tractor trailer or these Marine Corps kind of vehicles that you see here, could easily fit in the back of that and manufacture this with nothing more than a couple generators and some sterile water. And we aim for this to be fast and distributed, very adaptable and flexible. We can kind of take it, pick it up, and go where we need to go, so it can be fully deployed, and we don't have to be entirely centralized. When we envisioned this program, we envisioned places like the Democratic Republic of the Congo, and we envisioned places where we may be involved in stabilization or humanitarian missions. What we didn't envision was using this in, places like New Rochelle, New York, where if we had this device and we were able to make thousands of doses of vaccines in under a week, which is the goals that we're aiming for this program, could we have potentially put this in hot spots early on in a pandemic and stopped a pandemic and essentially stopped transmission before things got out of hand? Now, anybody that's familiar with the regulatory paradigms associated with manufacturing is probably laughing right now. And I fully understand that there are a lot of regulatory components here that we have to address. Of course, we need to ensure that the box is making exactly what we say it's making every time, making them of high quality, making them sterile. We have all the ability to test everything in the way we would in those very large facilities. So we have our work cut out for us. We're excited about the possibilities here, and also recognize that this isn't going to be likely a device that's sitting in your local CVS in the next 5 years. But I think there's a possibility that this could be sitting in your local CVS or at the local hospital in 10 to 15 years. And that's kind of the appropriate, I think, place for DARPA to be. The one last thing I'll say about this device is that it's very much focused on nucleic acid technology. So we really did double down from DARPA's perspective on nucleic acid technology, because we do feel that it is so much easier to manufacture. It's just a much simpler modality. And I think that there's the possibility that we could make with these devices, like I said, anywhere from 500 to 1,000 doses of vaccines in a very short days-long time frame. And You could envision a situation where you make many of these systems and you deploy them around the country to potentially respond to hotspots or infectious disease threats of the future. But of course, that the DoD could also take with them on the go when needed. And so this is where I think we can potentially take this in the future to allow us to respond even faster than what we currently do. So I think with that is kind of the overview of what we're doing here at DARPA in our pandemic response portfolios. I see that some Q&A and some chats have popped up, so I'm excited to have some conversations and answer some questions. But thank you all for your time, and it's been a lot of fun to tell you about this.
Thank you, Amy. That was a fascinating presentation. I know that as I was listening to you, I was thinking about some of the work that I've done, and one of the startups that I worked for back in the mid 2000s was actually funded under a DARPA program to do accelerated manufacture of pharmaceuticals.
Yep.
So, I know that you build on the shoulders of others when you join DARPA as a program manager and take on some of these programs. And I'm hoping that in the Q&A we can get into some of that, sort of how the sausage is made from the program manager's perspective. But let me pull up the Q&A here and make sure that I address the questions that our audience has put in. One that seems to be getting some votes from others as well is, as in your awareness, right? We know that you may have some limitations in this research as well. It's not your own research necessarily, but would a conflict occur if an RNA vaccine was followed with a vector vaccine like the Johnson & Johnson vaccine? Do you know?
Right. I think we don't know. I think that those are very good questions. So different technologies, I suspect that there wouldn't be, but this is just my personal opinion. And as you heard in the introduction, I'm a chemist, a biochemist, more than an immunologist. So I wouldn't claim to make any claims there. But I think just based on the fundamental technology, I think that there's likely not to be any adverse events from having kind of both modalities. And I'm sure that there will be studies to look at what happens when you have one vaccine and then you boost with another, and it may even become required, because it's going to be very difficult to track everybody for many years to come, which vaccine you got. So there would certainly, I would imagine, be investments in understanding that. A related question that may or may not have been asked, but something that we have certainly thought about at DARPA, is I mentioned the antibodies that you can give right away, and they're very protective immediately. Well, then the question becomes, okay, if you give that to somebody right away, would their vaccine work if you gave them the vaccine at the same time, or you gave it at a delayed response? And that is a question that we are very interested in understanding, because in an ideal setting, what we would do is we would give 2 shots, one in each arm. One gives you your antibody, one gives you your vaccine. The antibody works immediately. The vaccine takes 6 weeks. By the time the antibody isn't working as well anymore, the vaccine is. And so you've been fully protected that whole time. But there are very fundamental questions, particularly if your antibody would prevent the vaccine response from occurring. And those are fundamental questions that we're answering right now in both animal models and potentially even following up with some clinical data.
Thanks, Amy. And I didn't mean to put you on the spot with that question. I know I for our audience too, we all know we're flying the plane while we're building it, right? In this in this scenario. So we're learning a lot as we go along. And I'm just so pleased that DARPA, representing all of us, is out there on the front lines and doing these really types of courageous and risk-taking research that none of us could individually do, but we can do through DARPA. So it's fantastic. So the other question kind of leads into our discussion kind of leads into that. I mentioned that I was with a startup that was funded by DARPA to do this kind of work. And one of the questions is, you mentioned that DARPA funded Moderna early on. So is that type of funding from DARPA exclusively available for research, or does DARPA participate in funding startup companies?
Yeah, so DARPA is, I think, an equal opportunity funder in that realm. So we typically fund a variety of different types of institutions. So we'll fund small startups, we will fund small biotechs, we will fund pharmaceutical companies. I mean, AstraZeneca is a performer in our program. We will fund academic labs. We will fund, we do have small business initiatives. So I think I manage 8 small business programs. So we do efforts with exclusively small businesses. I will say, we have you generally break down the DARPA portfolios into our large programs like the pandemic prevention platform, and then smaller efforts that we may call seedlings, or things like our small business initiatives. So typically smaller businesses would have difficult time being a prime performer on a large DARPA program, because we typically ask the world of everybody and their large consortiums. So typically on the larger, it doesn't mean it can't happen, and it absolutely could happen. But typically on the larger programs, it's usually a mid-sized company that is either the prime, or even sometimes academic labs that have a lot of groups that are their subcontractors. And so where a small business would typically work under a large program would be as a subcontractor. And so, but that also is never tried and true. And there are times when we have large companies that are subcontracting to academic labs. So there are kind of very creative ways to meet our goals. And I think that that's one of the fun parts about DARPA, is that we do this work to meet a capability. The NIH plays a very critical role in the R&D ecosystem to understand science and to understand basic science. That is not what DARPA does. DARPA invests in science, we invest in technologies, but we want the ultimate capability in the end. So for example, in that last program I told you about, there's a lot of science that has to go down into that box, essentially, to make it work. There are a lot of investments, and there are a lot of scientific rabbit holes that you could go down. But we're not going to let them go down. We're going to say, okay, that's great. Now you've got a wonderful question you want to ask. You save that, and you're going to figure that out in some other time. We are going to drive towards making this capability. And so I think that it makes for a nice kind of funding arrangement, in that we are very much focused on that end prize. We don't necessarily care how we get there. And I don't mean to say that we're trying to be sloppy. What I'm saying is that if something isn't working, it's very important to understand why sometimes, but sometimes we don't care why it's not working. It's just not working on our timeline. Let's try the different approach and let's go. And our contracts and our approach can be flexible, and that we are able to very quickly shift. Maybe you incorporate some new subcontractors that can help you with a different approach, and you go down a different path to get there. And so I think that sometimes is the reason why DARPA is able to be successful and really push forward on some of these technologies.
That's hugely helpful insight, Amy. I think I had actually written down a question for you in case the audience didn't take us there, or you didn't take us there in your comments, but about looking at small businesses and university labs, which are two very different animals. What does it take to succeed as a performer? And you call them performers for a very specific reason. You mentioned that, phrase in your presentation as well, that the companies and labs that get involved in these projects are performers because they are supposed to produce something in the end, right? And
Okay.
so your insights about, what it takes to succeed, and that you have to stay focused on the target, not go off on down any other rabbit holes. That's very helpful for us, I think. I'm curious, as you're talking about this, how do the folks in our audience and others become aware of these programs and funding opportunities as they're coming online? You've just started the NOW program and others. How do we become aware of that?
Yeah, and this is really important because we really do want to reach, kind of other performer groups, because we know there are great ideas all over the world and all over this country that we want to hear. So one of the easiest ways, and it sounds a little cliche, but we have an opportunities page. And what we do there is anytime that we are having a new program that has been announced, we hold what we call proposers days. It's essentially an industry day, but we don't want to make it sound like only industry can come, because anybody can come. And we hold these proposers days for new programs. And this is your opportunity to come and hear from the program manager's mouth. This is what I want. Do not give me this. Give me this. This is what I want. And you can ask questions. Sometimes you can ask very pointed questions that the program manager is not able to answer, but oftentimes it's a great opportunity to be able to really understand what the DARPA, what DARPA wants. Additionally, what it allows groups to do, most importantly, in my opinion, and this is the most important part of a program, is that I would venture to say that 90% of our programs, they're at least within the biological technologies office. When we put out a call for proposals and we say, this is what we want to build, there is no one lab, no one company, no one place in the world that can do that. You almost always need your friends and family to come along and help. And so this is the opportunity, because you may look at this and say, wow, I couldn't do that. I'm not even going to think about it, but I could do this like one little nugget. Great. Go to the proposers days and find the other people that have the other nuggets, because oftentimes these consortiums, and as you saw on my slides, the Vanderbilt University and AstraZeneca collaboration was post facto. They were all performers on the same program and they came together during the pandemic. Oftentimes it takes different groups coming together to solve the problem. And that's, I think, really what DARPA is aiming to do. We do not expect that any one group can solve the problems. We want everybody's cool ideas to come together. And so you may not be a prime on a program, but maybe you have the one component and that's critical to the program and can participate that way. But I worry sometimes that people look at the metrics and the things that we set out and they say they're far too hard. There's no way I can do that. That's okay. Talk to other people and maybe you can make a contribution. And so those proposers days are the perfect place to do that. And we do have opportunities websites on the DARPA page that list those proposers days when they're coming out.
It's almost like you saw my notes in front of me, because my very next question was, can you tell us about what a proposers day is and what happens there? Having been on that side of it, I recognize how important those are. And then also that the aspect that you just mentioned as well, that once you get into these types of programs, you start to meet other performers and things might click that lead to even bigger projects down the road together and collaborations down the road. So very helpful.
Yeah, and we hold program reviews as well. So once we have a program ongoing and you may have, 4 prime contractors, but you may have 20 subcontractors, we bring everybody together to talk and kind of discuss the program and discuss their technology, usually twice a year. That's kind of waned a little bit during the pandemic, but I have seen some very wonderful collaborations come out of that. Collaborations that I wouldn't have even thought of. And I see 2 people talking and then somebody approaches me later and they're like, why didn't you introduce me to this person? This is going to be great. I think that's one additional advantage, is that oftentimes you just, as a researcher, and I kind of was there in my, my lab career, you get very focused on your technology and your kind of general circle. And it's great sometimes to get out and talk to people that have a completely different viewpoint. We like to try to bring together some of the high, an example is that we've liked to try to bring together some of the high performance computing and the computer science folks in the national labs with some of our people that are doing all this mining for antibodies, because there's a lot of data there. And so they may not be traditionally groups that would interact with each other, but they get together and they have this like great time just talking about large data sets. So it's been really, really rewarding to see.
Fantastic. I want to make sure that I cover some of the other questions that are landing in our chat. So I want to ask you, it seems as though serious outbreaks have been increasing in number over the past 2 decades or so. That's been predicted by many. Roughly when do you think we might be looking at the next scare? What is your thinking and DARPA's thinking, perhaps?
Yeah, it's a very good point. And just the time that I've kind of been thinking about this, we've had H1N1 in 2009, we had 2014 Ebola, we had Zika virus, chikungunya virus, another Ebola outbreak, and now SARS-CoV-2. Oh, and MERS, we have MERS in there. And then even back to SARS-1 in the last decade. So many that we can name now. I'm not an epidemiologist, and so I don't have a good answer for why this is happening. I mean, I think a lot of people, assume that, we are in closer contact with animals and just we're generally moving into their space, and that's just causing the opportunities. We also, there's more of us, right? We're close. And so we're closer together. There's the opportunity for things to spread. So, but I think epidemiologists could certainly than I could. When do I think the next one is? I don't know, but I would say prior to SARS-CoV-2, if you had asked me what the pandemic was going to be, I would have said influenza. And so I think all of us thought pandemic flu is probably the next thing to keep an eye on. I think there's going to be a lot of focus on coronaviruses for the next half a decade or decade, but I think we still need to keep our eye on influenza because it's still quite a significant threat.
I think we do. We all need to keep our eyes open for all of these threats. And I'm just so glad that, again, that programs like yours are ongoing and you're thinking up new programs every day at DARPA and working with your colleagues and collecting all of us from around the country to work on these big, hairy, audacious problems. Thank goodness. So with that last question, I think I'm going to wrap up the program. We're at our time point here. So I want to make sure that I thank you, Amy, for your presentation today. It was absolutely fascinating. And thank our audience for attending and taking time out of their day today to be with us. We are about to go into what's called our general networking session. You're going to be randomly placed at tables, those of you in our audience, in the virtual ballroom, and you can move around simply by double-clicking on another table that you'd like to join. There will be a list of attendees that are going to be posted to the right of the ballroom floor in the same box as the chat and Q&A features. You can just navigate. I'm pointing to my screen like that's very helpful. But there is a tab for participants right next to chat and Q&A. And you can figure out where everybody is, which tables everybody's at. So the entire event is going to conclude at 1:30. And I'm getting a message that there's something in the chat, but now I can't find it. So the entire event is going to conclude at 1:30, and I want to thank everyone for joining us on this Tech Tuesday event today, and we are going to move our presentation to the conversation mode and get to know each other. Thank you very much, everyone.
Thank you.